one of my PhD students now has a talk here at EHA presenting some of our next aims in this field. So we have basically, to make the story simple, we have been able, as I was saying, to characterize the evolution of the CLL from time of diagnosis to time of progression, relapse, transformation, because we have samples over a period of 20 years. Now we have tools that allows us to go even back in time. So we can reconstruct how the tumor has been evolving even before the diagnosis of the disease. And we do this and others in the field.
Welcome to EHA Unplugged, the official podcast channel of the European Hematology Association, EHA. I am Isabel Olivera, your host, and we have today the pleasure to have with us Dr. Ferran Nadeu.
Thank you very much for the invitation. It's my pleasure to be here today.
Thank you for being here with us. Dr. Ferran Nadeu is a researcher from the Molecular Pathology in Lymphoid Neoplasm Group in Barcelona, in Spain. He was awarded an EHA Junior Research Grant in 2021. Mm-hmm. And we are here to learn about how this grant has supported his career development. And to learn what your results have been, we're interested to see. Dr. Nadeu is a computational biologist and a molecular pathologist. He's specializing in hematological malignancies with particular expertise in chronic lymphocytic leukemia, CLL. He has developed new bioinformatics approaches to understand cancer evolution. So we are going to talk about your project that was funded by the EHA grant.
Perfect.
Can you please begin by explaining what the aims of that project were?
Yeah, so we decided to study chronic lymphocytic leukemia from an evolutionary perspective because it's a very intriguing disease. In some patients, the disease rapidly progresses, while in others, the disease is a very slow disease with a very slow turnaround. And this heterogeneity before therapy also occurs after therapy. When we treat the patients, there are some that respond for a very long time, others that rapidly progress. So we aim using different multi-omics technology, genomics, transcriptomics, to understand which is the biology behind these different clinical patterns. And this is more or less the general aim of this EHA grant that I conducted.
So your project aims to decode evolutionary pathways in CLL. What do you mean by an evolutionary pathway?
It's what is the biology behind these very different clinical courses. And in the first couple of years of my EHA grant, we focus on what we call Richter transformation, for instance, which is how the CLL cells, which is a small cell leukemia, mostly indolent in many patients, transform into a very aggressive disease. So understanding this kind of evolutionary pattern is that what we call like the evolutionary pathways of CLL. We could understand how this dynamic process occurs within a time window of more than 2 decades, understanding which are the early genetic alterations that are occur and how these cells change and evolve during all this decades of evolution of a tumor, also giving us the opportunity in the future for early detection of these aggressive clones and these kind of things.
So what is special in your approach to answer these questions, to understand this evolution?
So I think that what make our project special is that we have been in our hospital collecting samples from the patients and storing them in the freezer for more than 20 or 30 years. So this gives us the opportunity to analyze the sample, for instance, at the time of Richter transformation, which occurred at 2020, let's say, but we had samples from 20 years before. And we could analyze how were these cells at time of disease diagnosis and follow the changes during all this period of the disease. On the same patient, we had like 6 or 7 or 8 longitudinal samples collected over a period of more than 20 years. Mm-hmm. And this gives us quite a unique scenario to really understand how this evolution of the disease works at a genomic level and transcriptomic level. In our studies, we try to combine the different omics layers. We usually start with the genomics because it gives us which is the genetic drivers of the disease, because we know that in CLL there are some genetic drivers, so gene mutations, copy number alterations, that are associated with aggressive disease, others to more indolent disease. So we have the genomic information as a background, on top of which we want to build on the other omics layers. For instance, once we have the genomics, we add the transcriptomic to see how these genomic alterations might modulate the phenotype or the transcriptomic landscape of the cells. And in some of these studies, we also add epigenetic layers, DNA methylation, and ataxia, which basically gives you the idea of how the chromatin is packed or open in the nucleus, so which enhancers may be active in the cells and so on. So we aim to try to combine all these different layers to really understand which is the features that dictates this different clinical path, because we have seen that in some cases genomics alone is not sufficient to understand this process, but that there are some genomic bases, and then at the later stages there are some transcriptomic changes that is what at the end dictates these different evolutions.
So you're also using computation analysis in a special way. Can you explain what you are doing?
Yes, so we try to make sense of how we can integrate this data, because it's quite straightforward to do genomic analysis or transcriptomic analysis or epigenetic analysis. The problem is how we can integrate all this data. I have to say that we are still working on that, because it's a field that is evolving so fast, and there is every year new algorithms to do so. So it's a constant process. But what we have tried to do was to try to find in the different omics layers that we have some features that can be combined. For instance, in the whole genome, we have the copy number alterations to say, okay, these are the genetic alterations that are present in a subset of the cells. Then when we have the transcriptomic, using single-cell RNA sequencing, we can also identify copy number alterations there. And then we can kind of map each cells carry these genomic alterations with the transcriptome and link genomic and transcriptomic information, for instance. And when we look at the epigenetic information, we can see which are the transcription factors that bind in these regions and which are the genes that are more close to the different enhancers that we find, for instance, and is a way to link transcriptomic or RNA sequencing data with epigenetic information. So we try to make this kind of cross-omics integration of the different layers, and now we are working on algorithms that can allow you to put all of this together to have a much granularity in the analysis.
So you are developing your own algorithms?
In some of the analysis, we develop custom algorithms to try to answer some of these questions. We are also lucky that this is a field work in which many people is working on this kind of multi-omic integration, especially in the context of single cell. So we are also reusing algorithms that other colleagues from the community have developed.
So what have you observed? What can you tell us about what you have seen?
I think that one of the main findings that we did during this EHA grant was what we published about Richter transformation, in which patients nowadays have a very poor outcome. There is a much unmet medical need in this context. And this is a very aggressive transformation, but when we did this reconstruction of how this evolution takes place within the patients, we found what we call the seeds of this transformation up to 20 years before the clinical manifestation of the disease. So we now start to understand how this very aggressive transformation at the clinical level works or develops at a biological level, which may not be that aggressive because it takes up to 20 or 30 years. So this has basically changed quite a bit the paradigm that we had about how Richter transformation occurs. And this opens also a possibility to try to develop tools for early detection of this aggressive Richter transformation, and eventually maybe at some point to start thinking on early intervention in patients in which we may identify these early seeds at an earlier time point. And in addition to this more philosophical or evolutionary perspective of Richter transformation, we have also identified a lot of metabolic changes in these cells, transcriptomic changes that may guide to develop potential new therapies for this group of patients, that now other groups are also following this research line that we have started here.
So how can this work translate into better diagnosis or prognosis? Will patients be treated differently earlier?
Our genomic and transcriptomic characterization has allowed us to identify which are the genetic alterations that are associated to Richter transformation, for instance, or to aggressive course of the CLL. So this can help us to incorporate the analysis of these alterations in the clinics to identify which patients might be more prone to develop Richter transformation or to have an aggressive disease. And we are working now on integrating this genomic information in clinical models, in which we want to combine clinical variables together with genomic information to try to better predict how a patient at time of diagnosis will evolve, if the disease will progress faster, if it may have higher chances to develop Richter transformations. I think that from a diagnostic point of view, it may help. I also think that identifying which are the mechanisms that lead to this aggressive transformation of CLL may also identify potential new vulnerabilities of these cells. And we have validated, for instance, in our study that by targeting the metabolism of the Richter cells, we can stop their proliferation. It was a proof of concept to show that our findings may lead to the development of other drugs, but this is something that our group is not working on directly, but we hope that the community can take some of our findings and take advantage of that. And as you said, also from the concept of early diagnosis of these aggressive clones, we have shown that this can be indeed detected years before their clinical manifestation. I have to say that we still don't know exactly if we can find these aggressive clones in patients that do not develop this such aggressive transformation. Is something that we are still working on, that we wanted to finish during the EHA grant. It was not possible because all these kind of things always take a bit more time than what we initially expect, but we are working on trying to identify if this could be a marker of which patients will develop. We know that in those patients that will develop, these seeds are there, so we could aim for the future an early diagnosis. And for instance, we are working on developing single-cell technologies and algorithms that can identify robustly these cells early in time. But we still have to make sure that this is a real biomarker of this transformation.
So you were one of the ones who could fund the research with an EHA grant. How has this transformed your career?
Yeah, I think that it was instrumental to be able to conduct the postdoctoral fellow and together with my PhD mentor, because in Spain it's quite hard to get, or even impossible, to get a grant to do the postdoctoral fellow at the same lab that you did the PhD. But I think that this was the right idea to continue the research line that I started, because we have all the sample availability, we have lots of international collaborations. So it was like, for me, the perfect environment to conduct the research. And the EHA grant support not only my salary, but also give me the opportunity to have some funding to start creating my own research line a bit independently from my mentor. And this, I think, that is something that has been really crucial to help me apply for additional funding in the last years. And I have been lucky to get some additional grants in the last period of my postdoctoral fellow to start kind of setting my own track on this field of genomics, cancer evolution. And also I think that an EHA grant, it's an important milestone in a CV for its prestigious and as you said, it's not a grant that many people can get. So I'm very honored to have this opportunity. So I'm very, very grateful for that. And also not only for the EHA grant, but I will also try to take this opportunity to mention the computational biology training in hematology, that I could also participate in the first edition of the CBTH, and was also a very nice experience to extend my collaborations and my network of colleagues. And also was a very nice experience to keep growing and as a scientist, but also from a personal side.
Are you still participating in CBTH?
I participated in the first edition, and it was very nice because I could present some of the work that we were doing, some of the ideas that we want to follow now in the next projects, and I get extreme useful feedback from all the mentors, excellent mentors, all of them. And it was very nice because all the mentors were super involved in the projects, with a lot of feedback. And with many of them, I still in contact and collaborating. In addition to the EHA grant, which was crucial as well for my postdoctoral fellow, but also these mentoring programs of the EHA are very, very important and very useful for researchers that are starting their own research lines in this field.
So what comes next? Are you going to become a mentor, or what comes next for the project in general?
So for the project, we have one of my PhD students now has a talk here at EHA presenting some of our next aims in this field. So we have basically, to make the story simple, we have been able, as I was saying, to characterize the evolution of the CLL from time of diagnosis to time of progression, relapse, transformation, because we have samples over a period of 20 years. Now we have tools that allows us to go even back in time. So we can reconstruct how the tumor has been evolving even before the diagnosis of the disease. And we do this, and others in the field do this kind of thing using single-cell whole genome sequencing, in which you can sequence the whole genome of individual cells, and then you can reconstruct a phylogenetic tree of how these cells have been evolving, just like the phylogeny of the species or this famous tree of Darwin. So you can do the same for CLL or for any cancer. And this gives you super resolution to understand how these tumors evolve even before the diagnosis of this the CLL in this case, but in any other tumor. And we are applying this technology to better understand the process of Richter transformation, to really know when CLL starts, where the early alterations of Richter transformation occur, to better have an idea of which is the timing of this disease. Also aiming for this better understanding of which is the time window in which we could eventually develop early intervention strategies. And in the context of CLL, we are having now this talk here at EHA in which we have found that some patients don't have a single CLL, so a single tumor, but have multiple independent CLLs, which is quite fascinating from a biological point of view. And we are studying this to see why this happens in some patients, how these different tumors evolve during the lifespan of the patient and grow like in parallel, which are the features that are in common in the different clones, what make them different, and all this from this more evolutionary perspective of how these tumors evolve over the course of the years of the patient, even before the diagnosis of the disease, because we think that this, as has been shown in other neoplasms, knowing how the tumor evolved in the past is also very important to understand or to predict how this will evolve in the future. So knowing the past, we can better predict the future, let's say.
So what would you tell other young investigators that are thinking about how to fund their research, are thinking about applying for EHA grants?
Yeah, I would really recommend to apply. I know it's hard to get. I was privileged to get one of those, but I really think that they are all the support that you get from the EHA and all the other activities that we have mentioned, CBTH, but there are many others for non-computational biologists, the TRTH and others. It's a unique opportunity to extend your network, or some of them become then colleagues and collaborators, friends. So it's really, I think that all these initiatives from the EHA are crucial for young investigators to expand a bit or to go outside of their area of comfort, grow personally, scientifically, get input from top mentors. They have basically changed a lot the way I understand how to do science, or to have, say, a much broader perspective of how to tackle a problem, how to collaborate, and all these kind of things.
So if you want our audience to remember just one thing from this podcast, what would it be?
Well, maybe from scientific aspect, our work on understanding the evolution of CLL, especially on this Richter transformation, and the paper is out, published. So I encourage them to read. If they're interested in this evolutionary perspective of Richter transformation, I think that this would be the highlight of my EHA grant from a scientific point of view. From more a personal experience, I will strongly recommend to all the people that is my stage or a bit younger to apply for EHA grants, to participate in the mentoring programs that are available, that are really, really useful to keep growing as scientists, extend network of collaborators, and I think that they are very, very useful.
So thank you very much, Dr. Nadeu, for being with us today.
No, thank you. It was my privilege to be here.
And thank you to the audience for listening, and please stay tuned for more episodes.